Hello, everyone, and welcome. Again, may I remind you this session is recorded and also the following discussion. They will be uploaded to the YouTube channel, to our YouTube channel, and to the Facebook page, where you can already find the fascinating lecture by Professor Rosamund McKitterick from the forum's first meeting. I'm happy and grateful to welcome you all here today for the second meeting of the Israeli Forum of Early Medieval Studies. It is my absolute honor to present Professor Emeritus Patrick Geary of the School of Historical Studies at the Princeton Institute for Advanced Study. I'm certain that most of you are well familiar with his work, to which such a short introduction simply cannot do justice. Professor Geary is a leading historian of the early and central Middle Ages. His original and meticulous analyses of a vast array of religious, social, cultural, and political issues have become a fundamental part of the modern study of the European Middle Ages. He has directed the Reichenau and St. Gall Plan Project, an internet-based initiative for the study of Carolingian monasticism. Currently, Professor Geary is co-principal investigator of the HistoGenes project, along with Professors Walter Pohl, Johannes Krause, and Tivit Arvida. This extensive project studies the migration of European societies through DNA analysis, and we will hear much about it today. Professor Geary earned his PhD in medieval history from Yale University. He has taught in many leading institutions, such as Princeton University, the University of Florida, and the University of California, Los Angeles, to name but a few. And among many other lectures and seminars, Professor Geary has also delivered the Israeli Historical Society's 15th annual lectures in memory of Menachem Stern. Many of his publications have been translated to different languages, and they include, among numerous others, Furta Sacra, Thefts of Relics in the Central Middle Ages, Before France and Germany, the Creation and Transformation of the Merovingian World, and The Myth of Nations, the Medieval Origins of Europe. Today, Professor Geary will speak about HistoGenes, introducing genomic data into early medieval history. Since we are so many people, I kindly ask you to message me your name if you have a question. And now, without further ado, Professor Geary, the floor is yours.
Thank you very much, Asha, for this overly generous introduction. Let me see if I can share my screen with you and it doesn't crash the entire system again as it did earlier. And let's see, here we go. What I'm working on now is very different from what I have done in the past. And I am therefore pleased to have an opportunity to introduce you very briefly to this project. This is that we have just celebrated yesterday the first year of a 6-year project. So I'm afraid I don't have very much to tell you about startling results, but I would like to tell you a bit about what we're doing and the problems with what we're trying to do. Was I muted the entire time? Oh dear. Well, that's very bizarre.
Just for the past few seconds, you were.
Oh, okay. For some reason it muted. So shall I start over? Or what I'd like to do today is very briefly introduce you to this project that we are just beginning. We're 1 year into a 6-year program, and to present some of the characteristics and some of the challenges and some of the dangers that what we're dealing with. This is a 10 million euro project. We have 4 principal investigators from 4 different countries. We have over 35 collaborators coming from 9 different countries. We have 12 institutional participants from 5 countries. And our goal is very modestly to produce a comprehensive pluridisciplinary analysis of the Middle Danubian region from the 5th through 9th centuries. This is the area that we're looking at. It's the area of Europe with the most population shifts. And our written sources talk about all kinds of so-called peoples in this region: Pannonians, Romans, Goths, Huns, Longobards, Gepids, Bulgars, so forth. We have for this region over 100,000 excavated graves, many of them properly analyzed and published. And at the end of this period that we're looking at, from the Central European region emerged many of the peoples or nations of Europe that we know today. These are the areas and the cemeteries in which we are collecting our samples. As you see, Germany, the Czech Republic, Slovakia, Austria, Slovenia, Hungary, Romania, Serbia, and within these, we have a range of cemetery periods, some that run through the entire period, others that are used for only part of the period, so that we can look at developing communities on a local basis as well as more regional areas. Our objectives are to look at mobility in early medieval populations, but also to refine methods applied in archaeogenomics for historical periods, which is a major problem, and create an integrated multidisciplinary workflow. And this has been a problem for a long time, plaguing people trying to use genetics for history, that they tend to work very poorly with historians or archaeologists, and more of that later. What do we want to get? Well, we want to clarify the population history of this region from 400 to 900. We want to be able to assess the impact of migrations, political shifts, and cultural changes. And we want to analyze kinship, social hierarchies, networks, sex, gender, diet, social status, health of the Central European population. So that's the content results that we're aiming for. But at least as important is to refine methods to clarify population history in this historical period, but also to enhance the epistemological process. What do we mean when we use the terms that we use? What are the commonalities and the difficulties, the deep history of this kind of research, and what are the problems of talking about the genetic, historical, archaeological materials together? And then we want to develop new approaches to historical interpretation of genetic and archaeological evidence. We don't want to do, in the words of Guy Halsall, use 21st century methods to answer 19th century problems. We want to develop new questions that could have been answered, maybe not even phrased, prior to the development of these new methodologies. Now, there have been a lot of studies of ancient genetics, as I'm sure you all know, some very interesting work done in Israel. But if you look at this chart, you can see that almost the vast majority of these have been done in the Neolithic and the early Bronze Age. Very, very little attention has been drawn to using genomic data to study the period of the last, say, 3,000 years, especially the last 1,500 years. So these technological advances have made it possible to look at genomes, but they don't concentrate on our period. And part of the problem is that they don't really know how to integrate the very rich written as well as archaeological material of, say, the early Middle Ages into these genomic analyses. Too often, as one geneticist put it, geneticists make up stories to explain their data, or historians and archaeologists look for genetic data to justify the stories that they have made up. So with all of the current interest in genomics, books by David Reich, Who We Are and How We Got Here, or Die Reise unserer Gene by Johannes Krause, my collaborator, there is a wide popularity of genetic studies, I think excessively so. And the arguments that genetics will tell us who we really are, I find deeply troubling. I think the idea that genetic data will replace written and material evidence is very dubious, but how will it supplement? These are the questions, and what are the dangers? And there's a recent book by Misha Maya and Stefan Potzold, which is basically a frontal attack on Johannes Krause and this kind of genetic work. I've done a recent short monograph that looks at the challenges, but also the dangers of trying to integrate historical and archaeological material. Well, what is the problem? Why is this something that immediately makes historians and some archaeologists very nervous? And I think that the problem is very well presented in this image, because this is the first time that people have tried to integrate biological, historical, and material evidence to have a broader picture of the world. In fact, this is exactly what was attempted in the first half of the 20th century, particularly by Gustav Kosina, shown here in 1915 with General Field Marshal Hindenburg, delivering a lecture on his settlement archaeological method in the service of German nationalist ideology. But this is not a German phenomenon alone, widely accepted by archaeologists such as the Australian British archaeologist Gordon Childe and others. And this kind of settlement archaeology is still with us. What was Kostina doing? He assumes that every people, which he understands as a unique, homogeneous group, defined by a material culture, by specific physical characteristics, had a discrete homeland, which could be identified archaeologically. He understood, as did sociologists of his time, that a people shared a common culture, language, political structure, social identity. And his goal was to try to integrate sources in order to find the early history of these peoples. By tracing the material and biological evidence, he assumed that it was possible to follow pre-written evidence of migratory experiences of peoples. So this is just one example from his Ursprung und Verbreitung der Germanen vor und frühgeschichtlicher Zeit, showing the homeland of East Germanic peoples, a linguistic category which somehow he thinks is going to be going to manifest itself in material culture and in skeletal evidence. So he looks, for example, at a region with a particular material culture. He looks at skull shapes from the same area and assumes that people with skull shapes of this sort produced this culture, spoke the same language, and formed a people. And then he takes it beyond this. He's not content simply to work with ancient skulls, but assumes that modern biological types can be used as evidence of these populations in the past. So by looking at present biology, he can project this into the past. Of course, all of this is building toward racial science. And as he says, racial science achieves greater insofar as it not only reveals the physical, but also the mental and spiritual peculiarities of today's breeds. That is, the meaningful differences of the races. Well, I think that everyone in Israel knows where this is going. And the results of Kosina's work, which were foundational in Nazi racial ideology and attempts to create a not only a sense of modern races, but project them into the past, was a terrible inheritance, which was largely, but not entirely, abolished in the ashes of Berlin in 1945. Unfortunately, as geneticists begin to examine ancient DNA, many are afraid that they are revivifying the ghosts of Cosina and others who tried to do this kind of work. This is a paper from 2003, so in genetic history terms, ancient history, that objects to these critics who claim that it could be dangerous to link DNA with ethnicity. But they say it's very hard to understand why politics should be allowed to place constraints on academic research. I think it's very obvious why the politics of academic research need to be reflected upon. But there's a kind of early enthusiasm for some geneticists in the possibility of solving the riddle of ancient Germanic migration and so forth. So in 2003, this gentleman hopes that it's possible that the question of the early homelands of migrating Germanic peoples can be resolved. This is really answering 19th century questions with 21st century methodologies. That was 2003. And unfortunately, some geneticists are doing exactly that. This, for example, is a publication in a reputable scientific journal looking at Goth migration. Now, of course, these people are not referred to as Goths. This is a series of material culture complexes, but nevertheless, it is thought that it's possible to trace the specific genetic signal of so-called Goths from prehistory, from the following Jordanes' fairly mythical account of Gothic migrations, and connecting them genetically and material culturally to a series of complexes, and thus really doing very much what Cosina was trying to do. The danger then for our project is, given this dark history from the first half of the 20th century, and the fact that we are dealing with all of these labels that I showed you earlier, Pannonians, Romans, Goths, Huns, Longobards, even Sarmatians, Gepids, Avars, Bulgars, Slavs, are we going to be trying to find these labels and then connect them with a genetic signal? And this is what people are afraid of. And I understand fully their concern that once more, scientists not thinking about the past and historians, or rather positivist bend, are going to unite and try to replicate this kind of pseudoscience. Should we assume that there's a material or a genetic basis for making these distinctions? Well, how do we avoid replicating the dark past of such integrative studies? The idea is great. They were not doing genetics, but they're doing what they could, which was biology. And we're trying to integrate this kind of data. One answer is don't touch it, don't deal with it. I don't think that that's an acceptable approach. This is data. The question is, how do we tame it? How do we work with it? What are its limitations and its possibilities? And I think that first of all, we have to create the right team of people to work on this. We need to reflect on the subjective components of identity formation. We have to ask the right questions of our data. We need to recognize the limitations of genetic data, just as we recognize the limitations of historical and archaeological data. We need to develop a workflow for analyzing the data. And we must constantly dialogue across these very, very different disciplines, as well as with a wider public that understandably has been seduced into thinking that genetics will tell us who we really are. So our team, Walter Pohl, Austrian Academy of Sciences, one of the leading figures in the study of early medieval peoples, the nature, the complex meaning of ethnic identity and ethnic strategies. Johannes Krause, really the leading geneticist who is working in ancient DNA in the historical periods in Europe, about the Max Planck Institutes in Leipzig now, previously in Jena, whose laboratory is preparing and analyzing this material. Tiva Darvida, who is the leading archaeologist of this region, working in Avar archaeology, but more generally throughout the Danubian region, and myself. I'm not quite sure what my role is, perhaps conceptualization, coordination, a bit of the generalist working to bring all of the different areas together. So we really spend a lot of time on this, and this is a real problem. What are the subjective components of identity formation? Well, pots are not people, as is often said. Material culture does not define a people. And certainly genetic commonalities do not define a people. Identities are subjective cultural creations, and we all have to agree on that. One can find population groups based on genetics, but that is not a people. One can find morphology of material culture that may be characteristic of a location, of a region. This is not a people. We really have to understand identity or strategies of identity as a very complex process of at least three different elements. How one identifies oneself, but that's not enough. Acceptance of that view that one's identity is belonging to a group, but then whether or not that individual accepted as a group, but then also how wider society identifies the individual and the group. All of these are based on characteristics, but they are selective, and they are all culturally and politically controlled, so that as our sociologists such as Brubaker have suggested, identity maybe it isn't even a good word to use. Rather, it's strategies of identifying. Identifying is a process. It's not a solid thing. It's something that goes on in dialogue and dispute and claims made rather than recognition of something existing in reality. So we really need to think very hard about what these labels are and what the labels that we necessarily use when we talk about haplogroups or groups sharing common genetic background. And we have to ask the right questions. So how did the way of life in these Roman provinces change as the Roman order disintegrated? Origin and competition of the inhabitants during these changes of rule. What kinds of population continuities and discontinuities can we recognize? What kinds of migratory events take place inside of the region and then between Central Asia, Western Europe, and other regions? And then at a local level, and something that we're very interested in, what about these small local communities? How are they organized? How do they deal with gender relations, marriage strategy, health, biological kinship as a clue to real kinship, to cultural kinship? Does this matter? Under what circumstances? How can we see this? Social status, cultural practices. These are the kinds of questions we're trying to ask. And we're at the same time very important not to try to answer certain questions. Can we tell who is an Avar, who is a Slav, who is a Gepid from our data? No, that will not be possible. What precipitated population movements? Genetics will not give answers to the questions of why. This is the data that will tell us something about the what. we're certainly not going to be able to answer big questions like, did the barbarian invasions in the Roman political structures of the West? That is outside of the scope of what we're doing. And we certainly are not going to get into the question of Europe's dealing with its current migratory crisis. Now, that may seem obvious, but given the regions in which we are working, Hungary, Romania, Slovenia, Slovakia, the Czech Republic, questions about present identity, present national boundaries, and the deep past of this region are extremely hot topics, highly politicized, and we must work very, very carefully within these countries because we need cooperation of government institutions, museums, bureaucracies that are very interested in this. There is nothing academic about the question of what did the Middle Danube look like in the 7th century. These are hot button topics in this area. So we must be very careful about what we're asking and about what we're not asking. And certainly the extent to which we must not try to either intervene in contemporary arguments about identity politics or allow our work to be manipulated and used by people who are very much involved in these games. Our workflow development, what we're doing is collecting the Petrous bones from 6,000 individuals. The Petrous bone is a very hard bone that is part of the inner ear. It's been found that this contains hundreds, if not thousands % more endogenous DNA uncorrupted than any other part of the anatomy. And this has been an absolute game changer in all ancient DNA studies. So we're collecting Petrous bones whenever possible from 6,000 individuals. It's the largest ancient DNA project that, as far as I know, has ever been undertaken. We are concentrating on the comprehensive study of whole cemeteries in our core region. This is something that has not been done before. Generally, geneticists will sample extensively from a wide variety of cemeteries, sometimes over hundreds, if not thousands of years, in order to model population changes and shifts. What we are trying to do is look at intensively at local communities, sample every skeleton in the cemetery for which that has endogenous DNA that has survived, in order to understand social complexity and possibilities within the society. These are drilled and powdered. The powder is sent to Leipzig for library preparation and capture. And what we're looking for is 1.2 million single nucleotide polymorphisms, so-called SNPs. These are points on the genome. These are variants which have been shown to be particularly indicative of population differentiation. This is a particular SNP array carefully developed by Johannes Krause and has been very useful in helping us to distinguish populations in the deep past as well as in the historical period. Then select samples are sent to the US for whole genome sequencing so that we can have a much richer view. 1.2 million SNPs is a lot, but still the whole human genome is about 3 billion data points. We're looking at a restricted port, but for about 1,000 of the best, we're going to try to do whole genome sequencing. And then the analysis is going on in Leipzig at the Max Planck and Stony Brook in terms of the genetic material. Our analyses. The types of analysis we're doing, there are many, but one very basic one is principal component analysis. This is taking our genetic data and looking at what are the first and second principal components that most explain the variation of these individuals. This has been proven to be a very powerful tool to differentiate material, and placed on an XY axis, with the first and second principal components. The individual samples organize themselves spatially, and that spatial organization has proven to be very accurate in modern populations to look at a European, create actually a map of Europe. We are taking our ancient materials, putting them through this kind of analysis, projecting them onto a map of Europe in order not to say that these people are from these areas, but to understand how they fall apart. This is an early study, a pilot study that we did. And here you see that the principal component analysis of a cemetery in Hungary, a cemetery in Italy, produced two clusters. One that were they living today would cluster to Northern Europe, and were they living today to Southern Europe, Italy. Clearly two very different kind of genetic backgrounds in this population. It doesn't mean these are peoples. It doesn't mean that they're from Northern Italy or that they are Northern Europe or from Southern Europe, but that there is a distinctive signal within their genetics. And we see this when we do admixture analysis. We look at the different components of a population. Each of these is one individual from the cemetery in Hungary, and using a procedure called admixture, we look at different modern reference populations to see how might we understand the difference. What are the components? Everyone is mixed. This population, the light blue would be modern Finns, the dark blue modern Europeans and Brits, the red Iberian, green Iberian, the red basically Tuscany, central Italy. And as we look at this, we see that there are very different percentages of these mixtures in this population, with some with what would be today a very northern signal, others a very southern signal mixed. None of these, incidentally, correspond to what a modern Hungarian population from the area would look like. And then the third kind of analysis we're doing is biological kinship. Obviously, this is not kinship. Kinship, again, is socially constructed. An individual may have a biological relationship which is ignored, they're not aware of, or does not play a significant part within organization. But it is a suggestion. It's one component when compared with location of burial, type of burial goods, and so forth, to see if biological kinship is recognized culturally within a community. Again, from this earlier study, this is a cemetery in Hungary, where we see a large biological kin group right in the center of the cemetery. Then around them, others with the blue represents again that signal of a type of genetic background. This whole group, with the exception of one woman, all have the same genetic signal. Then they're surrounded by others with that similar genetic signal. And then on the margin are these other individuals with a southern signal, so that we can find within this specific here 4 kin groups. This is a large kin group of 3 generations. Then mother, daughter, and so forth. So we're able to actually look to 3rd generation of kindred to see to what extent spatial organization in death represents biological connections. And in some of our cemeteries, we see this kind of clear grouping by biological relationship. And in others, there's absolutely missing. And this can be 10 kilometers apart. Every local community has its own cultural traditions, its own identity. And we must not forget that and rush to big analyses of the whole region. Then we're developing a workflow process. We're analyzing archaeological data, as I say, focusing on each cemetery as a unique community. We're creating an extensive database on each grave, including grave goods, burial form, depth, orientation, and so forth. GS mapping of each grave within the cemetery so we can look at the structure of these cemeteries. And we're by no means limited to genetic data. We're collecting anthropological data, sex, age, height, trauma, pathogens, general health. And we're also doing extensive isotopic analysis, strontium, oxygen, nitrogen, some C14. Strontium is very important in understanding mobility because one absorbs strontium from water and soil of plants and animals from the location in which one is born and lives in the first years of life. So by examining strontium signals in molars, it's possible to know whether an individual is indigenous to the location in which they died and was buried, or if they are exotic to that area. Oxygen and nitrogen tell us about the diets of these populations. And of course, C14 helps dating. So it's a confirmation of the cultural dating that is done based on artifact and grave types. So we're trying to integrate genetics, but also isotopic data, anthropological data, and so forth. And we're also analyzing historical data, systematic collection of contemporary Latin and Greek sources. Of course, this region has no indigenous writing material. Latin and Greek narrative sources, legal sources, and then critical analysis of the written evidence concerning this region and its inhabitants, all of course written from outside, from the Byzantine world, from the Latin world, the Frankish world, or Italy. And perhaps the most important thing in this project is our constant dialogue within the different areas. We meet every Thursday, geneticists, historians, archaeologists, anthropologists, to take each other through almost a course in genetics, in cemetery archaeology, how isotopic analyses work, what kind of questions our different disciplines ask, so that we are teaching each other what it is that we do when we do what we do. And this is a very important way of working together and understanding each other's disciplines, so that genetics is not a black box that we either reject out of hand because of our fear of the past, or that we accept uncritically because it's so-called science. And the same is true for history, analysis of text, explaining to geneticists how one reads a text such as, say, Paulus Diaconus, Gregory of Tours, Theophylact. What are the strategies of composition? What are the uses of vocabulary and rhetoric, so that they're simply not picking and choosing labels and taking uncritically the stories they tell. We have weekly meetings. Post-pandemic, we plan physical team meetings and working plenary meetings. We're trying to organize one for the fall if Europe can get its vaccination act together. And then dealing with the dissemination of the results, progress in publications, but also in fora such as we're doing right now. And should you want to follow our progress on this, you can see us at HistoGenes.org. So, and then of course we'll be publishing in scientific journals, and that has its own rhetoric, and that's a whole different topic of what you can and cannot put in the scientific article that must be read as carefully as any other kind of highly elaborate rhetorical constructed text in order to understand what is said and what is meant. And we're also producing more synthetic publications in historical journals and monographs. Is this going to work? I have no idea. This is an experiment. The ERC is interested in high-risk projects, and I suppose they gave us this because it's so risky. So we'll report back in 5 years and let you know what has happened, but I hope you'll be hearing from us in the meantime. this is some of our team, but as I say, there are many more who are involved in this. And with that, I will stop and entertain your questions.
Thank you very much, Professor Geary. Fascinating talk. I know I have quite a few questions, and some of the members of the audience also. Let's start with Jonathan Harold. Jonathan, are you with us?
Do you want to unmute, Jonathan?
Yeah, right. Sorry.
He can't.
Taking security very seriously. Here. Okay, Jonathan, you should be able to unmute yourself now.
Okay, thank you. Yeah, thanks for very interesting talk, Professor Geary. Actually, you in your final slides, you kind of addressed the question that I had anticipated, which was how particularly stable isotopic analysis was working into your project, or if it was working into your project, or whether your project was going to focus primarily on the implications of a DNA analysis. Again, the sense that I get from the outside looking in is that if we're talking about the movements, certainly of individuals, biological individuals, but also organic elements of material culture as well, isotopic analysis in terms of provenancing people and things has some exciting possibilities that I think genetic analysis might suggest, but can't really establish.
You're absolutely right. Unfortunately, isotopic analysis can't either. We don't have a fine-grained, detailed strontium map of Eurasia.
Mm-hmm.
So, for example, if you look at the Pannonian Basin, if you look at the Danubian region, we have very large areas with a very similar kind of alluvial soil, and then they're mountainous areas. But some of these are, 20 kilometers apart.
Mm-hmm.
And so an individual buried at our site, Solod, for example, we find no one in Solod is indigenous to the location based on the strontium, except for children. But where are they from? Are they from 50 kilometers away or 5,000 kilometers away? Strontium will not tell us that. And certainly genetics will not tell us that either. So yes, we can look at that. There are some parts of the world where the people at least think that the strontium signals are sufficiently studied on a wide scale area. The UK or Great Britain is one of those. But for much of the world, all you can say is indigenous or exotic. Would you agree with that?
Yeah, once again, I'm kind of playing catch up on archaeology. I'm trained as a historian, so that conforms with my understanding of where this information is coming from. I just say that based on the scope of your project, it's exciting that that it attempts to connect with all of the different types of new information, new text, if you would, that is becoming available to us through scientific analysis or scientific methods, as well as the analysis of text and material as well. This is the way we should be working, I think.
Well, that's what we're trying to do. The problem is how to do that without privileging one source over another or corrupting one source over another. One of the big problems in Pannonian archaeology is that for the 6th century, essentially archaeologists have taken Paulus Diaconus' Historia Longobardorum as a guidebook. And they have corrupted their dating of material based on what they know to be the fact because they have read Paulus Diaconus. So it becomes a vicious circle. We want to avoid that kind of contamination when we combine archaeological, physical, anthropological, isotopic, genetic, and historical material. But that means constant diligence.
Yes. Okay, thank you.
You're muted.
Our next question is from Professor Röckelein.
Yeah, my question was in the same way to the isotopic analysis, because this would be, or it could be, I hope it could be a tool for the questions of migration, gender, marriage practices, and things like that. But you already answered what are the methodological problems with the non-differences of strontium. So I see there we will not have too much results from this part.
But don't be too pessimistic. We actually do get very interesting information. For example, to return to our site in Hungary that we studied in our previous publication in Szolad. Yes, all of the adults are born someplace else. But when you look at the isotopic signal, we don't know where they're from, but the strontium of the men is in a much narrower range than the strontium of the women, which suggests a differential marriage pattern that suggests that women are coming from a much wider catchment area and marrying into a fairly narrow group of men. Now, we don't know where they're from. I doubt that we will find out, although it will be interesting when we've got 6,000 samples and many thousand strontium tests. But it's already confirming that women's lives in the migration period are geographically very different from men's lives. And we have seen this already in other studies that suggest a greater genetic variation in the, and one talks about admixture. One of the things we're looking at is not simply the total admixture of an individual, but when we have whole genomes, or even with our 1.2 million SNPs, to look at the mitochondrial and the Y chromosomal differences in order to see haplogroups of males descent and females, to see what components are coming from mother and father. So some very preliminary studies are finding that while an individual may have mixed ancestry, there is a tendency for the mothers to be from a somewhat different category in some cases, genetically, from the fathers. So this is very, very preliminary, but it's beginning to give the suggestion that, in fact, we are able to discern gender differences within this this material, and the stable isotopic analysis is part of that. Also diet. Do women eat as well as men? Usually in society, not. We will see what goes on in these communities. So don't be too pessimistic. I think that there are going to be some very interesting questions about gender. Also, just a very basic thing, we have all of our graves are sexed by anthropological analysis of pelvises, but we also have the genetic sexing. And in one of our sites, just going through, we find 4 mismatches between what the anthropologists thought the sex of the individual was and what the genetics demonstrate it is. So we're going to also have a higher evidence. We have not yet found a female skeleton with sword, spear, and shield, but who knows what we will find.
Thank you.
Thank you. Our next question is from Merle Eisenberg.
Hey, Professor Geary, good to see you.
Hey, nice to see you, Merrill.
One thing I was curious about is something you touched upon in the last slide, which is something I've thought a lot about, which is publications and how you aim to different audiences. And so it's really good to see that you guys are doing, different fields, as it were. But how do you deal with the problem that I'm sure, which is the types of questions that science journals tend to want to answer, and then to get them to take, more historical questions, and then to follow up on that, how do you then make sure that the wrong questions and analysis aren't reified in, say, public spheres?
This is an enormous problem. I don't know if everyone is aware of the rhetoric of scientific publications. First of all, they're very, very short. A 14-page paper is probably about as long as most of these top-level scientific publications are. And most of that is methods and detailed technical introduction. So what you can actually say in the framing of the problem and what you can say in your conclusions is a real problem. We're struggling right now with a first paper that is almost finished, and the science is fine, but how do we talk about the context? How do we define the questions that we're asking? So the geneticists have been working on this, and they've sent this paper to me and to Walter Pohl, and we are trying to figure out how, within the same allotted space, to make it both understandable to scientists, but also not a caricature for historians. This is an ongoing problem, and frankly, we have not found the solution yet. But when you read these scientific publications, it's infuriating because these issues that you really need 5 pages to discuss have to be in 1 sentence. And this is a constant problem with the rhetoric of scientific publications. They also don't like technical archaeological terminology if it's a genetics paper, but they're very happy with technical genetics terminology. And so I don't know how we'll deal with that. We're working on it. The other thing, though, that you point out, I think, is one must be very, very careful not to have a sentence that some nut group can take out and put into their blog. And we've had that already with our publications. So we have to be very, very careful with every sentence to make sure that we are not opening ourselves to easy misuse. There will be misuse anyway, but we have to be very careful about the language, and we are constantly being watched, not just by the blogosphere Nordic nuts who think that we've proven that the Longobards marched from Scandinavia to Italy. We're also being watched by politicians. And there are big issues that I'm not going to talk about in a recorded YouTube presentation, but very, very serious political issues that we must be very careful to monitor as we do this. Will we be able to change the rhetoric of publications in Nature and Science and places like this? I don't know, but we're sure going to try.
Our next question is from Hillel Applebaum.
Thank you. For a person with a very poor understanding in genetics, this was a very interesting lecture, opening a new area to think about. I would like to ask one question or two. First, you said you cannot, and you repeated it, that you cannot monitor migration of groups or the origin of groups that you're dealing with, but within those groups that you test, can you identify movement in the within the time that you're dealing with? Can you find groups that migrated from one point to the other?
Yes, I'm sorry if I gave the impression that we cannot analyze migration. What we can't say is that this group are avars in Central Asia, and they are now avars in the Danubian region. It's a question of labels, but we can certainly see biological relationships that suggest, say, Central Asian origin or Western European origin of populations. And we can see differential change within communities so that we are able to look at migration through the genetics and through the strontium. Where the Avar Empire, we know that the Avars are a group of Central Asians. Who they are, whether they're the Ruan or not, is a big debate, and we're not going to determine that a certain label that appears in Chinese or Persian sources is exactly the same people that arrived here. But we can certainly say that there is a strong Asian genetic component in the elite of this population that settled in the region between the Danube and the Tisa around 580, 590. We certainly know that. We can also see other groups that have a different Asian signal in that population, as well as Western. So yes, we can look at migration. What we have to be very careful about is this ancient question: are the Avars really the Huns? Are the Huns really the Xiongnu? Are the Avars the Ruan, or as the Byzantines report from the Turks, that they're the false Avars, that they just took over the name? These are not questions that we can answer because these are cultural labels, but we can certainly look at migration patterns. We've done that in Italy, where we see mid 6th century Italy, one genetic pattern by 600, very different pattern within a community with a very large new population whose genetic signal would suggest a northern origin. And then we can correlate that with kinship. They're only marrying within their own group for 4 generations. We can correlate that with a higher animal protein diet and equipped burials and so forth, and say, yeah, this is a new population. This is a migratory population of some kind of elite into this region. So we can do a lot with migration, but we can't say, oh, this guy's a Lombard, this guy's a Gepid, and this one over here is a Bulgar. You had a second question.
Sort of second question. As I said before, I have I'm very poor in genetics, but in the popular, I haven't read the scientific papers about that. I know that certain genetic research can tell a person you came from there or from here or from there, and again it relates with migration. And in my research dealing with Jewish migration in Europe, that could, I don't know if you don't deal with those areas exactly, but I'm thinking ahead. Can we identify maybe with further research where people came from? But I understand from you this is very, very difficult.
Well, it's certainly possible. Now, a lot of this kind of popular genetic 23andMe, Ancestry.dna, and so forth, or using in a blunt way the kind of tools that we're using. It was found by John November, who was my colleague at UCLA, is now at University of Chicago, that if you take a modern map, take a modern sample of genetics of Europeans from all over Europe, using principal component analysis, you simply let them organize themselves based on their own distance relative to every other individual, without any concern for geography, simply on a two-dimensional graph. What you get is a map of Europe. And so what he says is the primary distinction of identity by distance. The closer together people are, the more they resemble each other, which my theory is, to put it this way, people have sex with people that they're close to, which tends to be the case. And as you move out, there's no hard and fast lines, but he has this very significant map that's been repeated many, many times. And it shows, it creates a map of Europe without adding any geographic coordinates. So more or less, you can say that you have this percentage of your genetic background, this admixture charts that I showed you, that would suggest that a significant component of your genetic profile correlates most closely with this, that, or the other region. That works. What we don't know, the databases that they're using for these commercial products are proprietary and secret. So we don't know what their model population is. Is it self-declared? How do they come up with that? Someone, what if you said, okay, this is the population of Israel, or this is the population of the US? Well, what is that really going to tell you about the deep past of a community? Because these are immigrant communities. These maps will say you are X% German. Well, is that German as understood in 2021, or is it maybe German as understood in 1871, or in between? What is Poland? What is Germany? What is France? They're taking modern political divisions and establishing those as though those were genetic divisions. It's highly problematic, but it is possible to a certain extent to correlate distance. What one does find that if it's identity by distance, this is a very strong component. But in a time of population movement, the other very powerful factor is identity by descent. And there are other techniques looking at whole genomes to, in theory, in every generation, every point of the genome except the Y chromosome and the X chromosome can recombine from the mother and the father. But of course they don't. You have very long segments that are identical. So by finding long identical segments, you can postulate closer or more distant connections, so that it's possible to look at identity by descent as well as identity by distance and see which is the factor. And then in migration, this is where you would have very similar populations in very distant locations, which would then suggest a migratory event that has taken place. But it gets very complicated because who migrates? Whole populations don't tend to migrate. What kind of admixture events take place in the course of migration and so forth. But sure, briefly, yeah, it can work. And there's certainly very interesting studies on Ashkenazi in Europe and the deep background of these populations. And this is this is serious stuff that can be useful, but it has to be handled very carefully.
Thank you very much.
Our next question is from Zoe.
Hi, thank you very much for the talk. It was really interesting. I have a question a bit theoretical about how to write these archaeogenetic papers. Should I introduce myself? Oh, I didn't think about that. I'm a PhD student at the Center of Paleogenetics at Stockholm University. And yeah, so I've noticed sometimes that this paper, they have this general catch title and then a historical introduction, using a lot these cultural labels that you say we should try to avoid to use like too much. And then we reach the discussion and then it's like, oh, it looks like that, but we cannot assert that it is this population. Is that like the best solution for writing an archaeogenetic paper? Because in the end, like, should we not choose these labels at all? In the end, you still need to introduce. It's like complicated. What is your advice of how?
This is exactly the problem that we struggle with every single day. For example, in the examples I showed you, this blue group and this red group, the obvious thing to say, this is a North Central European group and a Southern European group. But because the PCA plot is separating them, doesn't mean that they're from those areas. Some say, no, we talk about group A and group B. And we should probably twist the PCA plot so it doesn't look north and south, because that's arbitrary. So make it east, and we'll just say there's group A and group B, which would be a more honest way to present it. And then people say, no, you can't publish that with group A and group B. Yeah. You've got to give them names. So what are we going to call them? And that it's also a real problem. If you want to publish this in +1, you could maybe get away with that. If you want it in Nature, it had better have some sort of POW, here are the whatevers. And now we have discovered the Urheimat of the Goths.
Yeah.
So there is a push that you often see in very fine scientific papers. And you get to the conclusion, and oh no, they've flipped through a textbook that's 30 years old. They said, these must be the Huns. And literally, there's a very fine paper that I'm not going to name that does that. And you look in the bibliography, and they've read a couple of old textbooks. There's no historian involved in the project. But if they hadn't done that, they probably would not have gotten it published at the level that they did. So there is an institutional pressure to come up with some wonderful, exciting information that the public can connect. No one knows who group A and group B are. But if you say, now we can show that this group are the Vandals, then people will connect to that. So it's a problem, and we're dealing with that right now. The other problem that you're aware of, that one uses data sets that have been generated by other scholars, and we have to do that, and we're growing the data set. Well, those other scholars have been very happy labeling these with all kinds of ethnic names. So what are we supposed to do when we're using a data set that someone else has, and the way they have labeled them is by ethnic group? And we may not believe in the ethnic labels, but what are we supposed to do with their data, and how can we talk about their data? Because we're not going to go and redo their study. This is a dilemma, and if you have suggestions, please email me, because we deal with this every single day. And the geneticists will say, don't worry, we don't mean these are ethnic groups. This is just a label we're using for this region, or whatever. But that doesn't work when it's read by an historian or an anthropologist or an archaeologist. The other problem is that when you drill into some of these, you find that their group is based on 7 genomes collected over 1,000 square kilometers over a 2,000-year period. And suddenly that becomes a population, right?
Yeah, no, that's not realistic.
But it's how it's done. It's a very young science, a very young science. And all of us who've been parents have seen what young children do when they're beginning and starting out.
Our next question is from Lee Mordechai.
Yeah, thanks so much for the talk. So my question actually fits very nicely into the discussion now. And it's a bit, I'd like to hear a bit more about how HistoGenes is going to do outreach. So I'm thinking about an outreach strategy or maybe even a media strategy. So I'm talking about how to disseminate the results, not among academics, not necessarily among academics through the scientific journals or the historical journals, but among a broader audience, which, as you've mentioned in the talk, would also be interested in these results. Now, I know scientists have a lot of experience in doing science outreach, but on the other hand, that science outreach may seem, and if it is, a bit more simplistic. It might not suit our needs as historians. So I'd like to hear from you how the historical side would be reflected in this broader outreach strategy or media strategy.
Well, this is something that we talked about that at our PI meeting yesterday. How are we going to disseminate this more broadly? And we don't yet know. You're absolutely right that communicating with the wider public is absolutely essential. After all, they're paying for what we do. Also, this is information that, as I've said, in these parts of the world is very, very controversial. And it is being manipulated for rather nefarious purposes by different interest groups. So I personally think that we have a moral obligation to be involved in these questions. The challenge is how to do it. Now, scientists are not very good at communicating to the public. We've seen that through the pandemic in our country. Your country, Israelis apparently believe in science. Americans, certain parties, not so much, as you may have noticed. So how to convince people that science is not perfect, but that getting vaccinated might be a good idea. This is a problem. The American actor Alan Alda is very interested in communicating about science, is very unhappy with how poorly science is communicated to the public, and he's created an institute at Stony Brook University, which is where we have our one of our key collaborators, to train scientists using improvisation methods used in comedy to communicate about what they do. And there are even scientific grants at Stony Brook that you can only get if you go through Alan Alda's course in improv. And I think that we need to look seriously at that kind of technique of communicating with the wider public. Many of us give lectures to not only to scientific groups like this one, but to broader interest groups. Johannes Krause tried to write this book, Die Reise unserer Genome, for a wider public. He got dumped on by 2 medieval historians who are very unhappy that he wrote a, quote, bestseller. They call it a bestseller over a dozen times in the book. Clearly, there's something going on there. So Johannes is trying to do this. My little publication in German tries to walk people through what we're doing. But we're going to have to face the issue of wider dissemination of our results to a much wider public that really understands nothing about genetics or history or archaeology, but has an interest. And we have to do that in a region in which we must be very careful. There is a program being developed in one of the countries that we work closely with that is trying to create a presentation to counter some of these nativist, essentialist ideological movements. We cannot be directly involved in that because of the very delicate situation that our collaborators find themselves in. We need access to public museums. We need access to skeletal materials maintained by government entities. We must be extremely cautious in how we engage the public in certain areas without falsifying what we're doing, but also if we cross certain fairly obscure lines, we may find ourselves shut out of access to materials, and some of our collaborators might actually find themselves in some difficulty. So these are very, very difficult questions. Fortunately, we don't have any results yet to share to the world. But when we do, we're going to have to figure out how to do that in a responsible way. If you have suggestions, please, please send them. Perhaps you've had more success than we have. So send me an email with advice.
Our next question is from Dennis. I'm sorry if I'm butchering your last name. Sakino Komenko.
Yeah, close enough. Thank you. Thank you very much, Professor Geary, for a very interesting presentation you just gave us. And my question probably warrants a lecture in itself. So I'll maybe ask only for an epitome of a maybe forthcoming lecture. In light of what you presented us today, in light of the genetic discoveries being done by your team, how does this sit with the historiographical tradition that you're part of, started back by Reinhard Venskos and then carried out by Herwig Wolfram and Walter Paul? How do we assess the concept of people and peoples in light of, for example, the genetic discoveries? Is it probably not better, is it probably not more correct to speak of communities that we give different labels to? Sometimes we would rather call them political nations, sometimes ethnicities and peoples. Take, for instance, the opening clause of the founding charter of your nation and country, we the people, meaning we understand ourselves as a self-proclaimed or self-reflecting community. And then we just select a marker that we believe is more or less representative of this community, be it our skin color, be it our language, be it our religion or descent, or sometimes we invent those traditions. So how would you assess the correlation or relation between the concepts of communities of different scales versus people or peoples?
This is a fundamental question. Thank you for posing it. And certainly in the work of the people that you have mentioned, the genealogy from Venskos and Wolfram and Paul, and in my own work, I've written about early medieval peoples as constituted. And the image within the Roman world, that what makes the Roman people in the legendary history of Rome, it is a people who are constituted by law, rather than a biological entity. And this, of course, is what I've written about and what they have written about. Then we take a look at a cemetery where we see a population with a certain biological signal only intermarrying among themselves for 4 generations, with a higher protein diet and certain cultural characteristics, living within a group, other groups that don't cross over. And this is something of a challenge to us, quite frankly. how are we to understand that? And frankly, when I began this, I thought that what we were doing was studying the null hypothesis. Will we see a genetic differentiation within these migratory populations or not? And I thought the answer would be no. I thought that we would just have sort of a vegetable soup of European genetics, which at a superficial or even fairly deep level is true. All Europeans very, 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 very similar. But there are still distinct differences. And what I find quite interesting is how these differences in some communities can endure over generations. And what that, so we need to think about that. Now, that's not universally true. The first site that we looked at in Italy, that's clearly the case. We've then recently been looking at Cividale, which is the center where the Langobard the kingdom has first settled itself, and there's a lot of very supposedly Langobard burials there. And as we look at the material culture of people that the archaeologists say this is clearly a Germanic burial, we find that they are their genetics is completely Italian. So are these people Langobards? Are they part of the army? Are they under certain circumstances Langobards and under other circumstances Romans? Do they move back and forth? Are there multiple identities that under they're part of the Exercitus Langobardorum, the Langobard army, but religiously they may be something else. In their family they may be something else, and so forth. So this is we see these mixtures, but then we also see these groups in certain places that endure. And what I'm beginning to think is that this is a question of culture and of class, that culture is powerful. There are times when one marries across groups, and there are other times that one doesn't marry across groups. And it's not necessarily a question of ethnicity or racism. It's a question of religion. It's a question of class. It's a question of how one maintains property within a certain group. And the strength of cultural groups that through generations can maintain a distinctive identity through intermarriage, and long enough going on, that produces certain kinds of genetic variations that are more common within that group than within other groups. So I think that what I'm seeing is that it's possible to belong to different groups, but it's also possible to see certain continuities. And thus far, we've got at most 6 of these villages that we've looked at in some detail. And we have very different answers to this question in each of them. And we'll see whether that Vinskus Wolfram tradition of the heterogeneous constituted a people as a constitutional rather than a biological thing is predominant or not. And what we find, for example, thus far, it appears that in the elites in the Thebar Empire, they're a pretty tight group. They're married within that. So one step down, it looks like it's quite different. Well, let's look at the royal houses of Europe. There's really only one. You know, they're all intermarried. Well, now they're marrying out and they're getting into trouble for it. But basically, everybody's a descendant of Queen Victoria, and they're all Germans. There's a very, very strong ideology within an elite to select mates for certain strategic purposes, and that creates a genetic profile. Does that help?
Well, I think it adds in complexity to the already complex picture that we have had. So I would be really much looking forward to the end results of the project, because I think it's completely mind-boggling and it revolutionizes the way we actually understand the early medieval period, which I also study here at the University of Gothenburg. I forgot to introduce myself. I'm a PhD student here at the Department of Historical Studies here in
But we
Western
may
Sweden.
all find, this is the exciting thing. We're not there to prove that Walter Pohl and I are right. We're there to find out whether there's a new dimension that can help us understand the complexity of past societies. And there may be some real surprises for all of us. Everybody is a mixture here. There are no pure anythings, I guarantee you that. But what that means remains to be seen.
Thank you.
Luckily, I'm going to use my privileges as a moderator to ask a question that might warrant a long answer. I wonder, this question has it's two questions actually. I wonder what were the guiding principles in choosing the cemeteries that you've chosen? And also, I understand that you probably have very little results yet to show, but I wonder if we can already see differences between, let's say, monastic cemeteries and lay cemeteries, and between monastic cemeteries in different areas.
Well, the second question is easy to answer. There are no monasteries. This is a world, the world of the Avar Empire. There are probably Christian communities within this of various types, Arian, Orthodox, and so forth. For the most part, this is a pagan world. So we're not looking at religious institutions. There is one very interesting grave from the 6th century that was found near the Lake Balaton, and in it is a lead scroll with a bit of Ulfila's Gospel on it. It's the earliest text of Ulfila's Gospel. Who this person is, what they're doing there, their genetics would put them to Italy. The burial materials look more like that of a woman. It's a very peculiar grave, and it also may be a mismatch between the bones and the material. So it's fascinating, and we can't do anything with it because it was not properly excavated. But otherwise, we don't have that. The bigger question, though, that you asked, how do we choose the cemeteries? In our pilot project, we simply wanted a very well excavated and studied site from the mid-6th century in Hungary, and a well excavated and studied site in Italy. Comprehensive site, fully excavated, and material available. And of the thousands, there were only 2. So they are not chosen scientifically. They're chosen because this is the best data. So there's no idea that they're typical. In fact, they probably are not, particularly the Hungarian one is strange in a lot of ways. It's fascinating, but it's strange. What we're doing now in the selection is this is a selection made primarily by the archaeologists who know the region intimately. And what they are looking at is first geographical distribution. Secondly, sites that are that have some continuity, because we would like to find some sites that are occupied in the late Roman period and continue to be used through the period, the Hunnic period, the Longobard period, the Avar period, and see what happens in a longitudinal study of certain sites. There are others that are chosen because they are characterized by certain material culture complexes that the archaeologists have given labels to, that much disputed about what they're all about. So what we're doing with the 6,000 burials and several hundred sites is asking the archaeologists to look at the core areas where we will get complete cemeteries that are distributed through the entire region, that have some continuity. But again, with archaeology, you're stuck with what exists, what has been found by accident. We're never dealing with a random sample of all of the people buried in Pannonia in this period. We're dealing with a sample of those who were accidentally found because someone was building a road or there was some kind of excavation. So we can never claim that this is a purely representative sample of a population the way you might do a modern population study. We have to deal with the hand that we are dealt by the chance discoveries over the last 100 years. And then we have to look at those for which osteological material is preserved. There's an extremely important site in Hungary that has given the name to a particular type of ornament that's been very much debated and discussed in Hungarian archaeology. They've lost the bones. Nobody knows where the bones are. They're probably sitting in some museum somewhere, uncatalogued or miscatalogued. We would love to analyze them. We can't. There's a wonderful Italian site that we wanted to do. Because of the pH of the soil, all the bones disappeared. So there's nothing there. So it's a mixture. It's going to our archaeologists, asking them to select these sites. These are primarily, as far as we know, the burial sites of nearby villages. But even then, we have one burial site. Is that the only cemetery used by that community? Can you estimate the population? You can estimate the population using that cemetery, but it might be the population for a subset of the community rather than the entire community. And one can do field surveys and try to find out, but one can't be certain. It's not an exact science, but then exact sciences tend not to be either, except maybe physics.
Thank you very much for this answer and for this lecture. I have many more questions. I'm certain our audience still have more questions. Very much looking forward for the publications. One more word about our next meeting. Our next meeting will take place in the end of May with Professor Michel Zaltzman. So this would be the long early Middle Ages. And that's it for now. This recording will be uploaded to, as I said, to our Facebook page and to our YouTube page. Thank you very, very much.
And thank you all for listening and for your questions. And please write me if you have any suggestions, criticisms, advice, and so forth. We want to be in dialogue as widely as possible, and wish as well as it goes through. And my best wishes to all of you in this time of pandemic and political unrest. Be well. Thank you so much.
Thank you, Patrick.
Thank you very much.